All right, well, welcome everybody to week 8, the final week of the Epidemics MOOC. And we've had a fantastic discussion all through the course. And this week we've been taking a broader view, looking at the topic of global health and public health. And for that we have Craig Cameron, who's from the Department of Biochemistry and Molecular Biology, right? It's a mouthful. I was excited to have Craig join us in this discussion, both because of your history in working on poliovirus, which is a huge global issue, as well as some of the new therapies and new treatments you've been working on in terms of antiviral therapies. And we've been hearing a lot about antiviral therapies in the context of the Ebola outbreak, but also in the context of previous large outbreaks like H1N1. And so we haven't specifically talked about antivirals in the course, I don't think. So could you just introduce what do we really mean by an antiviral?
So in general, an antiviral is a small molecule, a chemical entity that's capable of interacting with a protein produced by the virus and preventing that protein from functioning, and in doing so, you prevent the virus from replicating.
So an antibiotic in principle kills the bacterium, but viruses, as we've variously talked about, aren't really alive in the classical sense. So there's a lot of discussion about what that really means.
I'm not sure we want to go there.
Yeah. So do antivirals actually attack the virions, the particles themselves, or do they just interfere with the function of them?
So there are many stages to a virus life cycle. So starting from outside the cell, you have the virus particle that attach to a receptor on the membrane that facilitates entry. Generally, you have to have some mechanism to get the genetic material that's in the virion out so that the molecular biology, the molecular genetic system can be used to produce the viral proteins and then go on and have replication. So any one of those steps could be targeted for antiviral therapy. So you have examples with some of the earlier and some of those drugs that are actually being considered now for polio bind to the capsid, and they prevent the proteins of the capsid from coming apart to release the viral genetic material.
So the virus is basically trapped inside itself.
Exactly. Yeah. So that's one example, but flu, right? Right.
So how does that work? Is that a similar thing?
It's the same.
Tamiflu,
yeah, same sort of idea as to prevent the uncoding, as they call it.
Mm-hmm. And so do we need a specific antiviral for every virus in the way that we would need a specific vaccine for every virus?
So my mantra right now is to create broad spectrum antiviral therapies, because you really don't know what the next threat is going to be. And even when you look at the evolution of flu, right, we've gone from H1N1 now to H3N2 is what I heard at a meeting in Taiwan a few months ago. So if you tailor your therapy, if you have to tailor your therapy to the strain, you're not going to have as much flexibility. So it's nice to be able to go after the guts of a virus, conserve molecular processes like reproduction of the viral genetic material. And so antiviral nucleosides targeting viral polymerases have worked really well with retroviruses targeting reverse transcriptase. And we don't have a similar class of antivirals for RNA viruses that have been approved, broadly.
Right. And we've heard a lot of discussion in this course and in the discussion forum as well about the use of antibiotics and the selective pressures that are generated by antibiotics, and some concern about broad spectrum antibiotics because they're not targeting a specific organism and have the potential to create a selective gradient for all kinds of things, even non-target bacteria. So how much is known about what kind of selective pressures are created by antivirals? Is it the same? Is there the same concern about resistance evolution?
So, I don't think we have the same amount of fundamental knowledge for resistance in the case of viruses relative to bacteria. Most often, when, so you think about the constraints of most viruses, the genomes are quite small. Every amino acid matters. And so when you start the process of evolving resistance, right from the beginning, you have reduced fitness. And so we have a lot of clinical experience with HIV. But retroviruses are really quite different than most of the other viruses like Ebola, flu. Retroviruses can actually replicate without any of their viral enzymes being produced. So you take in retroviruses, you take this RNA genome, you make a DNA copy. That DNA copy is integrated, and now RNA polymerase II produces RNA. So if you have a virus that is inhibited by a drug, drug resistance can emerge passively, right? You can't have that happening with other viruses, RNA viruses. So the extent to which the drug-resistant phenotype would lead to the ability of the virus of the body to clear the virus, it's not really known. I mean, it's not really clear. There's not a lot of experimental evidence out there. Tests, I shouldn't say evidence, tests.
So I guess Tamiflu, that's one of the drugs that have been the few antivirals that's been used a lot. There might be others also, but so is there any evidence of resistance?
There, you can create resistance, but whether or not you're enhancing virulence, at the same time is the question. I mean, and that's the problem with the bacteria. In general, if you give the bacteria a growth advantage, then these resistant strains can't be treated. So just the ability of the bacteria to grow is the problem. With viruses, in many cases, right, there's this constant battle between the virus and the host. And so if you take the fitness down, the host may be able to win. And so how that all plays out is not really, I don't think it's clear at all.
I think viruses as well have more of an ability, if you think about individual genomes, one may be, the fitness may be decreased, but it often can borrow the proteins it needs from the other viruses that are replicating in the same cell. And so it's called complementation. And you can have a population of highly defective viruses that are still capable of replicating and being transmitted because they just swap the proteins around.
But the inoculum size has to be really high for that to happen.
It's very high.
Yeah.
Right. That's right.
So I think what the other part of that is there's replication, and then there's the disease manifestation. And generally, for manifestation of disease, there's so many bottlenecks that you have to clear that, again, I think this is one of the reasons why I think drug resistance probably for classical RNA virus isn't going to be as big of an issue as it would be for something like HIV.